The first speaker this afternoon is Professor Kathryn North, who's the Director of the Murdoch Children's Research Institute and the David Danks Professor of Child Health Research at the University of Melbourne. Kathy trained as a paediatric physician, neurologist and clinical geneticist, and importantly, she leads the Australian Genomics Health Alliance, an NHMRC-funded national network of 80 institutions around Australia, and she's also the Vice Chair of the Global Alliance for Genomics and Health. And Professor North is going to take us through, in the very small time of about 4 minutes, going to take us through the network and core capabilities that are currently set up to prepare Australia for genomic medicine. Thanks, Kathryn.
Thanks, Roger. Hi, everybody. I really wish I was in the room in Sydney. And we've heard some great talks this morning that are really demonstrating the value of genomic medicine. And what I wanted to talk to you about from a background perspective is the work that we've been doing over the last 5 years as part of preparing Australia for genomic medicine, which originated as a targeted call for research through the NHMRC. This is really a big health services research project because the goals were to demonstrate the value of genomics in the diagnosis of one or more disorders, but also to demonstrate the cost effectiveness and particularly practical strategies for implementation into healthcare. And just 3 things I want to highlight before we go into the panel session and to give you some examples for. And first of all, one important thing in terms of integrating into our federated health system was to develop systems and resources that would actually mean that we had a cohesive approach around Australia. And so Australian Genomics really focused on creating a national diagnostic and research network, with the two very much intertwined. Australian Genomics currently has 450 members and collaborating investigators, over 100 partner organisations around the country. And what you see here on this map is the 32 national recruitment sites we developed as part of the flagship studies, which were really demonstrating from the patient presenting into clinic prospectively how we'd apply genomic medicine into healthcare. The big feat of this was actually achieving an ethics and governance structure that linked all of these together, but it meant that we could take the time to approvals from 18 months down to a turnaround time of just a couple of months for each new study as they came on. The goal here was to really have a coordinated approach around the country in terms of diagnostics in genomics, and for us on the world stage to be a single cohort of 25 million Australians. If you want to read more about this, it was published mid last year in American Journal of Human Genetics. I think there's also been a real value in terms of creating this social network, and this is a study that Jeffrey Braithwaite, Janet Long and their team have performed, and it really shows how over the first 2 years of Australian Genomics that we increased those sort of collaborative links between medical scientists, geneticists, other medical specialists and researchers, and really built that network of connectivity which is growing to this day. I think the next big issue we have to address is making sure that, that our genomic information and medical data is linked and stored and accessed and shared responsibly and safely. So not only do we need to create networks, but we also need to look at a whole of system change as we're integrating genomics into healthcare. So we tend to think about genomic sequencing as the technology, but really what we're looking at here is how do you approach ethics at scale? How do we look at data security, data access? How can we also store, share and analyse the data sets so that they can be then reused by researchers and other clinicians for the benefits of human health? One thing that we have done through Australian Genomics is to make sure we're partnering globally to help answer these many issues, so that we're a driver project of the Global Alliance, as you've heard from Roger, which is all about effective and responsible sharing of genomic and clinical data. And this collaborative now involves over 600 organisations around 90 countries. What the Global Alliance does, and everywhere that you see the logo, has been to develop standards and interoperability so that we can start to think about sharing data not only between our states, but really with other countries, and that we're all speaking the same language and we're all doing it safely and securely. And so this is where, as a driver project, we've now adapted, and this is our prototype in terms of genomic data from clinical data to contributing to world knowledge around clinical variants, and then developing a genomic data repository that's been part of our core work in Australian genomics and in partnership with the Global Alliance. So Roger, I'll leave it there.
Sorry guys, we're just having a little bit of trouble hearing you on the stream. If there's anyone in the venue who might be able to activate that microphone, that would be ace. Thank you.
Yep, we're good.
Yep, that's great. Thank you.
Introduction again?
Yeah, start again. Yep.
Okay, I think we had a technical glitch there and you may not have heard my introduction of Professor Judith Cho, who was introduced this morning as our keynote speaker and gave us a very wonderful presentation. So Dr. Cho is the director of the Charles Bronfman Institute for Personalised Medicine and Dean of Translational Genetics at Mount Sinai. She's led the Bronfman Institute since 2015, and that institute includes a major biobank named BioMe, one of the most diverse biobanks in the world. And hopefully this time, successfully hand over to Professor Cho.
Thank you so much. Can you hear me?
Yes, we can. Thank you.
Wonderful. So I think that in terms of breaking down the barriers to precision medicine, Kathryn alluded to some of the major kind of barriers that exist. One is proof of value in selected situations that will benefit patients. Key to that is harmonizing the data across different health systems. And so one of the things that we're doing in Mount Sinai together with Emer Kenny and Nora Abu Hussein is our involvement in the eMERGE network, which is an NHGRI-funded network. And specifically what the purpose of this is to demonstrate the value of these polygenic risk scores in a short to intermediate term outcome and show the value of these scores across populations. I think a second major barrier is training the new generation of workforce individuals so that that are trained in the use and the application of genetics, and including both monogenic and polygenic scores in clinical practice. And the third major barrier that I see for the widespread application of precision medicine is computational in nature. The electronic health records, understandably, is a major platform by which we are trying to integrate these data. But beyond that, I think some of the other types of information, the new data streams, including wearables, including other ways of communicating with patients on an ongoing basis, and developing the kind of new either cloud-based platforms or app-based platforms in order to provide more immediate information going back and forth to patients represents another major barrier for widespread application of precision medicine. And I'll leave it at that.
Thanks very much, Judith. Our third presenter is Professor Ingrid Sheffer, who is the president of our academy and also the chair of Paediatric Neurology Research at the University of Melbourne and a senior principal research fellow at the Florey Institute. Professor Scheffer is a physician scientist whose work as a paediatric neurologist and epileptologist has led the field of epilepsy genetics for more than 20 years and has led, among other things, to the award of the Prime Minister's Prize for Science in 2014. And Professor Scheffer will now speak to us about precision medicine opportunities in paediatric neurology.
Thank you, Roger. And I hope my 4 minutes will be very complimentary to Kathryn and Judith's, where they've given big picture application of precision medicine. And I'm going to make a deep dive into the severe childhood epilepsies and just give you an example of quite how challenging this question is. So I'm talking about children who have frequent seizures, often every 5 minutes. They have abnormal EEGs with very frequent epileptic activity. And associated with this, they have developmental impairment and a whole host of comorbidities, so very profoundly affected children and adults. We now know that these diseases are highly genetically heterogeneous, monogenic diseases, and here you see a figure showing many different cellular biological processes are implicated. So let's just think about one of those, and this is a sodium channel gene. I'm going to speak particularly about SCN2A, which encodes the alpha-2 subunit of the sodium channel, which is a major ion channel in every cell in our body. SCN2A has been associated with a host of severe epilepsies as seen in this pie, and you can even have autism spectrum disorder without epilepsy. What's turned out to be really interesting is that the profoundly affected gain of function children have onset under 3 months of age, and they have a very poor outcome. With these other syndromes, they have loss of function mutations. And I'm going to drill down now onto this more severe, the early onset variety of the developmental and epileptic encephalopathy. And these children begin under 3 months of age. So looking at a gain of function mutation in these individuals, the obvious question for gene therapy is that you want to block that gain of function. And by that, we want to restore it to normal levels of function. But there is a challenge. We don't want to go too far, or we'll have a loss of function phenotype. And this could also have a very severe phenotype associated with it. The challenge is greater though. We have to get it into the right nerve cell, we have to get it into the right network, and we have to get it in at the right stage of development. Is that in utero? Is that under 3 months? Is it after that? So turning now to a mouse model developed by my colleague Steve Petru and his lab at the Florey. And they have developed a mouse with a recurrent variant that causes this early onset, very severe epilepsy. And they have made an antisense oligonucleotide with Ionis. And here in the black, you see the mutant mouse, and you can see they're all died by 30 days of age. And here in the blue and then the pink, you see the ASO given to that mouse, and at higher dose, a remarkable improvement. So this looks super exciting, but there are huge barriers still that we have to overcome at the research clinical interface. Firstly, mouse is not man. There's a chasm really between applying a drug in a mouse and then one has to go to safety and dosing studies in non-human primates, and then safety and dosing in humans. These are rare diseases, so we need sufficient numbers of patients at the right age to test the gene therapy, that we have to know they're going to be profoundly or severely affected. How do we monitor outcome? It's got to be a lot more than seizures. We have to look at all those comorbidities. This is going to require worldwide collaboration and implementation, and we've already begun to discuss the socioeconomic costs involved. And what a take-home message I think is really important is to work with families and communities so that they understand the challenges as well as the promise of precision medicine. Thanks very much.
Now I'd like to introduce Professor Chris Goodnow, who is the Executive Director of the Garvin Institute of Medical Research and holds the Ritchie Foundation Chair as Head of Immunogenomics Laboratory at Garvan. Chris has made many major contributions to science, particularly around immunology, and is a Fellow of the Royal Society and the Australian Academy. And Chris is going to talk to us about the promise of precision medicine.
Thanks, Roger. And thanks for having me in this virtual crowded gallery online. I really enjoyed the talks this morning, and I wanted to reinforce some of the messages that Judith Cho talked about in the context of autoimmunity. Let me see if I can make these slides go forward. How do I do that? There we go. Perfect. So I wanted to talk about precision medicine, particularly in the context of autoimmune disease, and I think it'll reinforce and complement a lot of the things that we've been talking about today. And to really put it in context, I just wanted to introduce one case, this little girl, Carla, who developed crippling rheumatoid arthritis at the age of about 15 months, very severe, and a journey that she was on with some of the best paediatric rheumatologists you could get, in this case at Westmead Children's Hospital, is this sort of, you can see here, this growing list of options, starting with the old standby prednisolone and methotrexate, and then a growing set of biologicals and small molecules. And the challenge then for a physician and a specialist is, well, which one do you use? And the rule book says, well, you try methotrexate. That wasn't an option in her case for some other reasons. She was on mycophenolate, wasn't really doing the trick. So she then started on the first of the TNF inhibitors, adalimumab, and that was, as I'll show you, doing something, but it wasn't really solving her problem. She was still in a lot of pain and was crippled. And but along comes this person on the right, Owen Siggs, and the other reason for telling this story is that it illustrates one of the barriers, I think. So Owen has a PhD in immunology, and so he really understands the immunology and genetics. He has done a postdoc where he learned how to do all the computational genomics, and then he's also got a medical degree. So in one brain, he has the whole package, and he made the diagnosis that Carla has a de novo mutation, a monogenic disorder, in the very famous break on the immune system from a cancer immunotherapy perspective, CTLA4. And this mutation completely disrupts this inhibitory receptor from binding its ligand and doing its job. And so what that meant straight away was that Carla and her physicians could skip the list and go straight to the targeted therapy that was going to replace her defective CTLA4, which is CTLA4 Ig or abatacept. Bottom line is that was a tremendous outcome. So here is a proxy, a lab proxy for her inflammatory state is an erythrocyte sedimentation rate, and you can see not much benefit from prednisolone and azacitidine. That's for another problem that she had. Adalimumab doing something, but then she goes on abatacept, and particularly one of the other advantages that this gave is it put her physicians together with physicians at the NIH and other places who see other kids with CTLA4 mutations who said, no, twice the dose and fortnightly, not monthly. And she's now 3 years out from that, and she's just this bouncy little kid who, when she came into Garvin, found all the science very boring and just wanted to find out where was the chocolate brownies. So the message there in that case is how do we connect genomic profiling to people who will best benefit? She's an extreme case of a monogenic disorder, and we've heard this morning about that we've still got ways to go for polygenic disorder. And of course, most kids with juvenile rheumatoid arthritis, and certainly most adults, it's much, much more genetically complicated. And the challenge is all the ones that Judy just, I think, outlined. How do we do that on the science side, but how do we get that into the hands of physicians that need to use this to make decisions? So the only other case then, it goes to the other extreme of this problem. still rheumatoid disease, and this is a person with a rheumatoid factor autoantibody, one of the first autoantibodies to be discovered, and one that's a great way to start an argument amongst clinical immunologists as to whether it has any utility in the clinic. But in this particular case, and in this disease, which is cryoglobulin vasculitis, the rheumatoid factor is the pathogenic agent, and it causes the pathology you can see there. But the question is why, and we don't have, or haven't in the past had the laboratory tools to make sense of it. It's all still a bit hocus pocus, and that's very frustrating for a clinical immunologist or a rheumatologist. But what's become possible in the last couple of years, and this is really, is to do single cell profiling of and find the cells that are making this antibody. And it turns out that what differentiates a benign rheumatoid factor from one that causes this disease is a couple of somatic mutations that just affect solubility of antibodies. And so you can turn a very complicated and mystical thing into something that's biophysical and very precise. Now, the other thing that's come clear by this possibility is that these rare cells have driver mutations, 1 or 2 typically, in most individuals with this disease. The cells that are making this antibody have also got mutations that drive lymphoma. And so that provides a potential bridge to bring drugs that are initially developed in lymphoma across to autoimmune disease. And again, I think that reinforces the message that Judy was talking about this morning, that precision cancer medicine does provide us with a lot of guideposts for how to do this in other diseases. But again, it's how do you connect the molecular immune profiling to people who best benefit? And, you know, how do you design molecularly guided clinical trials? So the final thing I just wanted to come back to is that list, because you may or may not realize, but this is a historic time for people that treat immunological and inflammatory diseases. We've gone from having the old standard of prednisolone and methotrexate, or dexamethasone for the president. Never felt better since the last time I was on dexamethasone either. But we've now got all of these small molecules coming through, biologicals coming through. Many of them are approved and have been shown to be very, very effective. And the real challenge is, how do we use all this technology and science that we've got and bring that together with this, what's now going to become a major challenge for every physician and for our health system, of which drug for which patient, and most importantly, how do you make it cost effective? Because here's the cost of a few of these antibodies. Rituximab is off patent, originally developed for lymphoma, but it's still $6,800 a year to the PBS, $41 to the patient. Abatacept, CTLA-4, what Carla got, $5,800. Tocilizumab, a newer drug for rheumatoid arthritis, and of course also the drug that's very important for CAR T-cell therapy to stop the cytokine storm, more expensive. And then the small molecule, one of the JAK inhibitors, tofacitinib, which does look really good in the clinical trials for rheumatoid arthritis, but with some rare but severe adverse events. But you can see, even though the cost of goods is probably a lot less than a monoclonal, the cost to our PBS is pretty hefty. So I'll leave it there, just to say those are really, I think, the opportunities and the threats.
Thanks very much, Chris, who's going to deprive us of the opportunity of a dexamethasone high. Next speaker is Professor Erin Alexander, who's the head of the Gene Therapy Research Unit at Children's Medical Research Institute and the Sydney Children's Hospital Network. And he holds an appointment as a Senior Staff Specialist and Director of Laboratory Research within Children's Hospital Westmead. Ian is a national and indeed an international leader in gene therapy, and there's nobody better to speak to us today about gene therapy, use of genes as medicine. Thank you, Ian.
Well, thanks, Roger, and a special thanks to the Academy for giving me this privileged opportunity to speak briefly. So in the few moments that I have, I want to obviously address gene therapy as a precision medicine paradigm. And this is a field that's had a protracted 25-year gestation, but has recently started to realise some tremendous real-world therapeutic success. And as we see those stunning developments moving from the preclinical stage through clinical trials to registration and to marketing, we're seeing the whole translational pathway being pressure tested. And I'm just going to try and very briefly share that with you. So in this first slide, I've attempted to capture the entire history of the gene therapy field. And along the bottom, I've listed the number of trials each year over an almost 30-year period. Then mapped onto that, I've tried to capture some of the challenges, some of the stunning successes, and the barriers at the research clinical interface. And I just want to work through those with you. So some 3,000 trials in nearly 40 countries over 30 years. And the first point I want to make is that between the first trial, which took place in 1990, and the first success in 2000, a whole decade passed, 400 clinical trials, no therapeutic success. And that really highlighted the first challenge, and they're the biological and technological challenges of actually getting to the clinic and having a therapeutic impact. And it really related to the underlying technologies that weren't adequately mature, but have subsequently matured and have started to deliver the therapeutic successes. And I've listed a few here that have been reported in the New England Journal of Medicine. The successful treatment of haemophilia B in adults using liver-directed gene therapy with AAV. The successful treatment of chronic lymphoid leukemia with genetically engineered T cells that we know has exploded into the entire CAR T cell field. And then more recently, stunning success in the treatment of spinal muscular atrophy, which is a lethal neurodegenerative disease of infants. And these, many of these trials have progressed through to registration, and many are starting to come into Australia as late phase trials or even registered products. And so they're starting to test our regulatory environment, and already we're starting to see issues. For example, CAR T cell technology, which is a grade 4 biological, goes according to the Australian regulatory guidelines for biologicals, but AAV gene therapy, where you go straight into a patient, is going down a pathway for prescription medicines. And that's probably not ideal, and there'll be issues down the track. The other thing that we're seeing is bringing these therapies into the hospitals, and that's happening. In fact, by Christmas, we'll have treated 21 kids with SMA here in Sydney, which is very exciting, but that's stressed the hospital system and is testing health system readiness. And the major challenge there is having the health system ready to be able to handle GMOs and meet the regulations under the Gene Technology Act. And then this explosion of global activity and many biotech companies flooding into this space has also created a worldwide shortage of recombinant viral vectors and gene transfer formulation at clinical level. And this is putting the field back 18 months to 2 years as we speak. And then there's some very dramatic logistic and commercial challenges. And you'll notice here that above each of these entries here, I've talked about gene addition. But when we look to the future, we're moving to an era where precision medicine is going to become even more precise. And so I just wanted to very quickly give you one example of what we've been doing in the genome editing space. And basically, we've been able to harvest livers from children with OTC deficiency, engraft cells into a chimeric mouse human model, and then genetically repair those cells in vivo at single nucleotide resolution, and do that at therapeutically effective levels. So this is the trajectory of precision medicine, but it raises some profound issues because there are 500 mutations in the OTC gene, and you're going to be able to develop bespoke reagents for each of those mutations. And what is the regulatory system going to do in terms of dealing them, and what are the manufacturing and translational challenges? So I'm going to just leave it there and just end with a few key issues and questions. And I think the logistic and commercial barriers are very significant as precision medicine becomes even more precise. And if we're going to realise these benefits, then we're going to have to work out how we can put them through the regulatory pathways at a lower cost. And so I won't go into all the questions that I've listed here, but I do think that the way our Therapeutic Goods Administration regulates this sector is going to be important, and this is true globally as well, and particularly in what the expectations are going to be about all of these bespoke therapeutics that are being developed for either a single mutation, and it's very much akin to developing these sort of therapies for ultra-rare disease. So I think we've got tremendous challenges in those two spaces, and I'll leave it there, Roger.
Thank you very much, Ian. Much appreciated. So now we've got a technological challenge. We're going to do a mixed real virtual panel. So I'm going to invite Ian and Chris to sit at the front. I can see our virtual panelists on the screen in front of me, so that's great. And hopefully our online audience will be able to see everybody in turn. We've got about just under 15 minutes for a discussion. I have no questions at all on the chat function. But I'm sure there are going to be plenty either coming in or in the room. I'd like to start off, since this is particularly around, and what we're trying to address here is the barriers at the research clinical interface. So I'd just like to go around the panel really quickly and ask you to nominate what you think the top barrier is at that interface. What is it that we need to work on? Number one. So let's start, we'll go in the order of the panel. So Kathryn?
I think the big issue we're facing is once you provide the evidence to demonstrate clinical efficacy, cost effectiveness of a genomic test in a specific situation, then how we jump from that into clinical care in terms of it being funded within the health system.
So essentially it's funding is number one issue.
Yeah, well, it's getting it through the, even once you've demonstrated that it's, it's cost effective, it's better, it gives you better diagnostic accuracy, there still is to go through MSAC is the traditional pathway, but that's not really going to be the appropriate pathway if we're to take a sort of a holistic approach nationally in terms of how we roll out genomics.
Right. So it's sort of the funding decision pathway. Judith?
I think the major barrier is workforce understanding. There's been a certain way that medical care, medical education has occurred. And this is a sea change, and it's being integrated into medical curriculum. It's being integrated along the kind of training pathway, but it's going to take time. We have to think fundamentally differently about training and medical education.
Thank you. Ingrid?
We're right now? Sorry.
Yes, technical
thing. To build on what the others have said, I think translation is a huge issue at many levels. And you said, assuming clinical efficacy, how do we get it implemented? But I think we'll have several different, even gene therapies to approach one problem. And we need to have very good standards for evaluation and for comparison between them, and then the cost issue. So I think it's actually incredibly complex and deep. So I would say that full translation question.
Thank you very much. Chris?
Is that working? Is that working? Yeah, workforce, again, I think is number one for me. Both the training, how do we, particularly as all the medical training in Australia has gone to postgraduate, that means in principle we have fewer and fewer people who are like Owen Siggs, who I mentioned, who really understand the science and really understand the medicine. And then even when you have those people, we've got a system in our hospitals where it's becoming harder and harder for someone to balance doing the research with doing the practice. In New South Wales, too many of our gifted people are visiting medical offices where they only get paid for the sessions they sit, and I hear that as a problem.
Ian?
I agree with everything that's been said, but in line with what I said moments ago, I think the regulatory frameworks that one has to negotiate to get into early phase clinical trials and the associated cost burden that comes with that, particularly when you're taking so many potentially bespoke therapies through that you, for relatively small target populations, you just can't sustain the traditional cost burdens that have been associated with regulation. So I think we have to reconfigure our regulatory system to really get to the precision of precision medicine, if you like.
Thank you. So I guess, so you've mentioned cost of the regulatory system itself. What do people think, how are we going to address the even greater costs of the medication and so on? And this is a question I'll put to any member of the panel. How are we going to make the whole precision medicine enterprise financially sustainable? Don't all rush to speak at once.
I think, sorry, I was just going to say very good health economics is critical, because you need to know about the true cost of a disease, and that doesn't just involve a sick child, but the parents not working and all those broader implications. So I think health economics would make a major step forward to understanding whether the cost of a gene therapy is worthwhile.
So sort of the cost to the whole of government, education, housing, all of the other social costs associated with the disease outside of, in addition to the cost in the health system itself.
Whether the parent can work, for example.
Yeah. Okay, look, there are a few, thank you for those. There are a few things coming in on the chat line. So one of them is a question for Ian. How do you ensure that new treatments are accessible in an equitable way?
I think this is an absolutely fundamental. This is an absolutely fundamental question, and I don't think anybody has got the answers here. And I think as we move towards increasingly bespoke therapies, the costs are going to rise, and therefore the challenges of access and equity are going to increase. And I actually think in, say, terms of genomic editing, which is an area I spoke to, we may actually have to back away from attacking conditions that are, say, single nucleotide resolution to whole gene resolution, and that might partly offset some of the challenges. But I think there are functional limits to the precision of precision medicine. And so I think some of the way we can defray costs is pull back from unnecessary precision and try and make sure that the translational pathways are not overly burdensome, and there'll have to be incentivisation for rare and ultra-rare conditions coming through from government.
Is there an optimal point at which health economics comes into this, or is there a phase in the development of new therapies where you really just have to work on the the fundamentals and getting the treatment right, or should we be thinking about health economics right from the very beginning?
I might comment on that, Roger. I can't see you, so just nod. I think it has to be built in from the beginning because there's so many phases that we need to look at. And certainly with looking at the application of genomics in different disease groups and then looking at diagnostic efficacy and cost effectiveness, it differs markedly for different disorders. And so it's also not one size fits all depending on the disease subgroup that you're looking at. I also think when we start to look at population-wide genetic approaches such as polygenic risk scores or pharmacogenomics, they will need to be assessed quite differently in terms of benefits in preventing adverse effects across the health system. That's a very different assessment to looking at diagnostic efficacy. So I think that approach we need to build in at all points.
So look, there's a question that I'm sure everyone will find extremely easy on the chat line. In all seriousness, it's a really key question, and that is, how do you balance the benefits and the financial implications of this very exciting things that we've been hearing about in these sessions with things like improving aged care?
Well, I guess one of the, I'll jump in there. I guess one of the things that we need to talk a lot about is preventive health. That's the hardest area to deploy genomics in because, statistics means never having to say you're sure. And, but if you take cancer again as a leading thing, that what ideally we want is precision targeted treatments that allow you to identify cancer early when it can be removed surgically before it's metastasized, as opposed to, triple highly targeted treatments in, stage 4 disease and, that buy someone an extra year or two of life. And so, I think really the history of real return of investment to society of medical research has been in allowing all of us to get to a median age of in our 80s, and with a bit of luck, we're actually pretty healthy most of that time. So I think there's a lot of work to be done there to figure out how do we get the prevention side of things there, because that's where the real returns will be.
So here's a really interesting question, and the questioner says, I asked this question earlier and Professor Cho gave a great answer. But if I can ask the rest of the panel, who is it who should be delivering precision medicine? And then a follow-up question, and it really gets to what you're talking about, Chris. How do we encourage more clinician scientists to stay in the system?
Jump in there. One of the things that I really don't think we want to do is have specialists that are genomic medicine people. I mean, you might need them, but just like every other technology advance, it needs to be integrated into each one of the specialties and ultimately into the GPs. A great example is celiac disease. There's very nice evidence that with a simple genomic profiling of the HLA that could be done for $30 for everyone, and then if someone has the at-risk allele, which is a few percent of people, you do one antibody test, which is highly specific. You could pick up the 2/3 of people that are never diagnosed with celiac disease until they develop severe osteoporosis in their 60s. So that's a GP level thing that should be implemented. But who's going to implement it? The GPs don't even understand genetics.
And keeping people in the system, what's the answer to that? Does anyone on the panel, how do we ensure that we have a growing workforce and not a diminishing workforce of clinician scientists?
I think in terms of developing quite specific pathways, and certainly the Academy has done a lot of that, and I think that there is focus around the country of demonstrating the value of the clinician scientists, supporting them immediately post-PhD, for example, where they'll often sort of drop off the cliff back into clinical practice, making sure that we're creating jobs that have research as part of them. And, that's a big ask, but I'm starting to see a lot of quite good models around the country. And I think it's also up to us to advocate so that the clinician scientist stream is also recognised within the investigator pathways within NHMRC and MRFF.
Can I jump in there as well and add to what Kathy said? Couldn't agree more. The Academy has got major mentorship program for senior people, but also a program to try and entice students, young doctors, into life as a clinician scientist.